this session to talk a little bit about the examination, a little bit about the format, but also to give you some example questions. And what I've tried to do in this session, or what I will try and do in this session, is to kind of give you some idea about the level of knowledge that you'll need to know. So quite often people will come and say, right, you might have looked at the screencast and you might be thinking, my God, there's a lot of knowledge here. There's a lot, it's very dense. How much of this do we actually have to know? How much, well, obviously you have to know it all, but in what kind of detail do we need to know it? So that's what I want to try and do in this session. So what I'm going to do is I'm going to give you some practice questions and you're going to discuss them amongst each other and you're going to come up with some answers, hopefully. We're not going to record any marks. It's, you know, a complete free hit. But also what I'm also going to try and do is point to the learning aims and how the questions fit around that, and also show you where the answers come from on the slides. And so hopefully by doing that, you'll get the idea about, you know, if we've got a slide that's very busy with text and lots of information, okay, what are the important things that I need to know? What level of detail do I need to know? How, you know, what am I going to need to know to pass the examination, obviously? We want you to read it all. We want you to understand it. But also, of course, we want you to know what the important facts as well. I haven't got my pointer, so I might have to stand over here and push the thing. Also, I'm going to record this, so I need to talk into my computer a little bit as well. So, Q&A on muscle and nervous system topics. I know that we did the nervous system first a week ago or so, and muscle this week, but we're going to do it in that order. Nerves first, then muscle second, I think. Yeah, so the first thing is the nervous system. So hopefully you remember that we posted, it was 3 hours worth of lectures into screencasts, hopefully in nice bite-sized screencasts, and hopefully you enjoyed the screencasts. So what I thought I'd start off by doing is just give you a couple of slides about the lecture aims for the neurophysiology content. So first lecture, maybe the first hour lecture, broken down, as I said, into these screencasts. What we wanted you to do is to understand the basic divisions and the organisation of the nervous system, the role of the central nervous system. So we talk about the peripheral nervous system and the central nervous system. We want you to understand about neurons. So neurons are the functional unit of the nervous system. We want you to understand the electrochemical basis of the nerve action potential. So that sounds a little bit daunting, maybe. What it really means is that we want you to, are you listening at the back there? What we want you to do is understand the electrical chemical basis of the nerve action potential. So we want you to know what ions are involved in the nerve action potential. That's what the electrochemical basis means, if you didn't quite understand that. We want you to understand about action potential propagation, and we want you to know about this thing called myelin. So essentially, when we write these questions as lecturers, we're told to take these lecture aims and concentrate on those as the type of questions that we're going to ask you. For the other lecture, we wanted you to understand what synapse is. We want you to understand neurotransmitters and receptors, hopefully all kind of basic stuff. But we also want you to understand something called synaptic integration and function. So a little bit more tricky, maybe. We'll talk about what synaptic integration is and synaptic function. Okay, sorry, next one. Final, there was 3 hours worth of lectures. So we want you to know about the divisions of the peripheral nervous system, so maybe the autonomic and the sympathetic nervous system. Something called proprioception. There's some questions on that. We'll see what that is in a minute. To understand the function of the somatic nervous system, to understand that the NMJ, the neuromuscular junction, is a specialized excitatory synapse. So we use the NMJ, the neuromuscular junction, as an example. Acetylcholine is released to get muscle contraction. And we want to know something about this process here called excitation contraction coupling. And hopefully you'll remember that when we do the muscle physiology, we also talk about excitation contraction coupling. So maybe we should be asking you questions about that on the examination, because it's one of the key learning aims. So yeah, like I said, what I'm going to do is I'm going to go through the type of questions that we ask you. So I'm going to give you about 5 or 6 MCQs, multiple choice questions, typical, taken from the bank that we use. Obviously, this very same one will not be on the exam. It might be, I don't know. But give you an example of the type of depth of knowledge that you'll need to know. So what I'm going to do is I'm going to encourage you to chat amongst each other for a couple of minutes about each of the questions, come up with an answer, then we'll give you the answer and we'll go through where we got it from in the lecture. Okay? So here's our first question for today. Which of the following is not a type of chemical neuronal synapse? So you've got 5 things there. Chat to each other, and I'll come back in about 30 seconds' time, and I'll ask you for an answer. So off you go. So what do you know about synapses? Okay, so anybody want to give me an answer? Someone up there, yeah? Yeah. Yeah. So which one of those do you think is not a typical neuronal synapse? You're right, that was a just nice description of a synapse, yeah, at the back there. One answer only. So which are you going for? B. B? 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 Okay. Any other answers? So remember, that's an important point. There's one right answer. So B and C is, I think one of them is right. B. Okay, I think lots of people are saying B. Yes, we're right. Good. Okay, so where do we get that in the lecture? That's probably the important thing to think about. So this is a slide taken directly from the lecture. And what you can see on this slide is basically, it's called neuronal synapses. And there's a diagram there that's got 4 different types of synapse labeled. So you could either look at this one and say, right, from there, I can see 4 of them labeled. I can't see B, gap junction. So the 2 ways in which you can answer this type of question, you can either know that gap junctions is wrong because I've told you or somebody's told you something about a gap junction, or the fact is that you can recognize the other 4 as being correct. Okay, so gap junctions is the correct one in this case because it's the odd one out. So again, like I was saying, if you look at this thing here, you can see that there are synapses between axons and dendrites, axons and other axons, axons and soma, and dendrites to dendrites, but not a gap junction. So a gap junction is actually an electrical coupling. It's used in lower animals, but probably not in humans. So you could have got that answer by either knowing this or knowing that gap junctions was the odd one out, essentially. This question is a little bit similar from that point of view. So which one of the following statements about an excitatory postsynaptic potential, an EPSP, is incorrect? So again, be careful when we ask correct and incorrect. So again, have a little chat and I'll give you 30 seconds or so. Okay, so what do we think? Anyone got an answer? Which is the odd one out? Which is incorrect? That's maybe a good way to think about it. Incorrect, odd one out. I don't know. Might help some people. Yeah, what are you saying down the front here? C or E? Is that what you're saying? I think one of those is right.
E, I think, okay, so it's not E.
It's not E. So the answer is, I think, if it's right, C. So why do you think C is correct? Anybody? I know few people said C. Why do they think C is correct as being the odd one out? What's C describing? Yeah, good. An action potential. So a bit like that first question, if I show you the slide that it came from, try and use my pointer. Whoops. So yeah, if you look in your notes, you'll see that there's a page on EPSPs, and it tells you that they're graded, temporal, and can summate. So graded, summate, and decay with time means temporal. So actually, that was probably, like you were saying down the front here, C and E are kind of the two that stand out as being incorrect. Now, they can lead to cell depolarization, but this is the other thing. All of these questions, you'll see this a bit later on when we've got a bit better examples of this. It's always the best answer. So actually, if we're writing multiple choice questions, what the GPHC wants us to do is write them that they're all correct, but one is more correct than the other. That's a bit too difficult for us to write questions for you to answer. So here, an EPSP could lead to a cell depolarization, but it doesn't have to. So again, maybe if we look at this, or we can look at some other diagrams. But the most important thing, as somebody said, was C is actually a description of an action potential. So an action potential is all or nothing. It goes to the same height all the time. So again, you could have either known a lot about EPSPs here and said, well, I know that A, B, D are not incorrect, are correct. Not sure about E, but actually I know that an action potential, which is different to an EPSP, is all or nothing. So again, like the first question, you could have either known that it was the odd one out by knowing about action potentials, or you could have known, or you could have worked it out by knowing something about the EPSP from this diagram here. Okay, now this one's a little bit more tricky. Which one of the following statements about the sympathetic nervous system is incorrect? So again, which is the odd one out? So have a look at those. I've kind of done this one deliberately. I think it is a little bit more tricky than the others. What do we think the right answer to that one is? Have a little discussion amongst yourselves for 30 seconds, a minute or so. So yeah, I can hear some people down the front here doing exactly the right thing. They're saying, well, a certain number, that's correct. That's correct. So I think the way to work these ones out is, if you don't know what the right answer is straight away, think about which, in this case, think out which ones are correct, and then you can rule them out. So there's a couple here that I would say you should know are correct. Maybe 3 that you should know are correct. And that probably boils down to a choice between 2, I would say, but I might be wrong.
That's where I'm at.
So go on then. Let's have a guess or a If somebody knows the correct answer. So which ones is it not? A, B, and C. It's not E. So that leaves us with D, correct. So, you know, what I would say about this one, I would say you should know that noradrenaline is the main transmitter in the sympathetic nervous system. You should know that acetylcholine is the main transmitter in the other system, the parasympathetic. So you can rule out B. You should know the sympathetic mediates fight or flight, whereas the other one mediates rest and digest. So you can rule that one out. It does innervate the liver because the liver is important in fight and flight. So that probably leaves you with a bit of a 50-50, I would say. Can you remember if it comes from the thoracic lumbar or the other one, which is cranial sacral? Or can you remember where the postganglionic fibers innervate? Are they close, distal, or are they far away? Sorry, are they far away, distal, or are they close? And actually, the right answer is D. That is the one that's incorrect. So the one that is incorrect is D, if that makes sense. And again, if you look on the slides, I tell you something about the parasympathetic nervous system, the sympathetic nervous system, and something called the enteric nervous system. And I also give you and I say there, the ganglia distal. Distal means further away, so that's the incorrect answer. And again, you can see it from this diagram where some of them innervate close and some of them innervate further away. A little bit more tricky to see on this diagram, I'll admit, but that's where we tell you it in the lecture. So again, you know what, as I was saying, the reason I left this one in, this is a little bit difficult. We might not actually give you this on the final exam, but it's a process of elimination. If you know that you should know what the main neurotransmitter is. You should know what the fight and flight versus rest and digest. You should know the liver is important. It was just those other two. And it was a bit of a 50-50 for me if you could remember which one was which. So here's maybe a bit more straightforward one that maybe you know, you don't know. It's not really testing anything as involved as the last one. So there's something called a myenteric plexus. Now, is that a neural network within sympathetic or parasympathetic nervous system? Is it in the somatic? Is it in the motor end plates, which is the neuromuscular junction? Or is it in the enteric nervous system? Have a little chat about that one. Does anyone want to have a guess at this one or tell me the right answer to this one? Maybe. So we have someone down the front here saying enteric nervous system. Does anybody want to disagree? Correct. Enteric nervous system. So let's have a look where we got this one from. So I tell you a little bit about the enteric nervous system, not a lot, but the ones that I've got two slides. This is one of them. And one slide tells you the names of the two plexuses, if that is the correct plural, plexi maybe. But the myenteric or Albach's plexus is part of the enteric nervous system. So again, you know, maybe you should know that it's not part of the motor end plate. It's not part of the, what was the other option? Oops. It's not part of the somatic nervous system, because again, that's to do with the motor end plate. Then you've got a choice probably between sympathetic, parasympathetic, and enteric. And again, hopefully you know a little bit about some of the nerves in the sympathetic and parasympathetic, and it's not those. So it's the enteric nervous system. Okay. So we tell you that neurons are important, but we also tell you about these non-neuronal cells. So which of the following is not a type of a non-neuronal cell in the central nervous system? So remember that non-neuronal cells are glial cells. And then we've got a choice of 5. Have a quick chat about it, and I'll give you 30 seconds or a minute or so. So let's have a guess on this one, or an answer. I shouldn't use the word guess. So we got D here at the back there. D. Good. Everyone's going for D. I think D is the correct answer. Well done. So I think this was a little bit like those first 2 questions. Let's have a look. I told you that there are some non-neuronal cells, including astrocytes, oligodendrocytes, microglia, and these epidermal cells. You'll see that Schwann cell is not there. So you could have, like we were saying for the first ones, you could have got the answer to that by knowing that the 4 non-neuronal glial cells are those 4. If not, you could have known that the Schwann cell is important to myelinate axons in the peripheral nervous system. So not in the central nervous system, in the periphery. So their Schwann cells would have been the correct answer. They myelinate axons in the PNS, the peripheral nervous system. Okay. So I think last one before we do some short answer ones. So the reason I put this on here is this kind of a bit of an annoying question maybe, because it's opened, closed, membrane depolarization, hyperpolarization. It sounds like it's easy to get wrong. Which one is the correct answer? So voltage gated channels, calcium channels in the nerve terminal, are they opened by depolarization? Are they closed? Are they closed by neurotransmitters? Are they opened by hyperpolarization? Or are they opened by neurotransmitters? So they're gated by voltage. Which one is correct? Anyone want to have a guess? Anyone want to give me an answer? Say again. D. We've got one for D. B. D. Okay, sorry, D. Anybody else? A. So we've got a few for A, a few for D. So what I would say here, this is one of the reason I put this one on here, and like I said, it's a bit of a, you know, open, close, hyperpolarization, depolarization. It might sound a bit like a trick question. The reason I put this on here is because a couple of these answers are correct, but one is more correct than the other. So actually, the correct answer is A. So you open the channel when you depolarize the cell. So if it's voltage gated, that means it's opened by voltage. And it could be opened by hyperpolarization, but hyperpolarization means you make it more negative. So I would say, but if you look at the other ones, so here, for example, there are neurotransmitters that close calcium channels. And there are neurotransmitters that open, but not all neurotransmitters open and not all of them close. Whereas if you depolarize the membrane, if you make it more positively charged, you will always open a calcium channel. So on the front of the paper, it will say, here's a bunch of multiple choice questions. Please select the single best answer. So unfortunately, if you wrote C in the exam, you wouldn't get a mark. If you wrote E in the exam, you wouldn't get a mark, even though technically they're correct answers. So it's a bit like I was saying before. What the GPHC wants us to do is give you 5 correct answers, one of which is more correct than the others. But actually, if you depolarize the membrane, you always open a calcium channel. So A is the single best answer. Yeah. And again, this was the slide. So this is a diagram that probably comes up quite a lot. So this is chemical synapses 101, if you like. You'll notice that in my lectures, I probably have this diagram up 2 or 3 times. And again, a little maybe exam tip. If you see a diagram that comes up more than once, it means that it's important. So again, if you don't know anything else, learn the diagrams that come up a lot. You'll see a bit later, I'm going to ask some questions that are based around tables. And again, as you know, when we're writing questions, if we've got a table with lots of information, it's easy for us to ask a question about it. So know the tables, know these diagrams that come up a lot. So you'll see here it says neurotransmitter release is triggered by the arrival of an action potential. And it doesn't actually say on here directly that it's a voltage-gated calcium channel that's open, but I talk about it in other places. But essentially, depolarization activates the system. Okay? And this diagram is going to be important, maybe as we'll see later on. So those were MCQs, 5 or 6 MCQs on the neuronal physiology. How about this one? So I'm going to give you a couple of short answer questions that are based probably on diagrams that are in the lectures. So have a look at this one. So the question here is, in terms of ion channel involvement, so I'm giving you a bit of a clue, we want you to know about ion channels. Describe the physiological events underlying the nerve action potential shown above. And hopefully you remember from the learning aims, one of the learning aims was to know about the nerve action potential. And we're going to give you 4 marks for this question. So the first thing, probably have a chat amongst yourselves again. The first thing to think about is where are these 4 marks coming from? And I would say here, if you've got a diagram that's got A, B, C, and D labelled, that's 4 different things. So maybe that would be a good place to start. So have a little chat. Think about how you're going to get 4 marks. What 4 pieces of information are you going to give me? When for the marker. And think about which ion channels are involved here, 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 and here. So I'll give you a couple of minutes. Yeah. Yes. Some channels are open by hyperpolarization, things like chloride channels. But in this case, if you depolarize a calcium channel, it'll always open and let calcium in. So I'll give you a couple of minutes on this one. So have a think what's happening in each one of these phases. Back to the learning aims, you know, which ion channels are involved in the nerve action potential. Okay, so what do we think then? Let's start with resting state A. It might not be the best place to start in this question, but what are the ion channels that are open when the cell is at rest? None of them. Yeah, good. So nothing's open there. What about in B? Sodium, good. So sodium channel's open, we get depolarization. What about C? Potassium, good. Harder. D, what's happening in D? Yeah.
Protein, try to bring back the
Good. Yeah, exactly right. So D is, you get this undershoot basically because the potassium channel stays open a bit longer than the sodium channel. And extra information, which was really good there, this sodium potassium pump kicks in and resets the concentration gradients. Okay, so where would you have got the marks here? Let's have a look. So if you'd have said ion channels are closed and there's no ion movement, you'd have got 1 mark, 0.5 for each, I guess. Probably if you'd have said nothing's open, I'd give you a mark. Here, sodium channels are opening, sodium entry, probably just giving you 1 mark for saying sodium channels, 1 mark for potassium channels. This was a little bit more difficult. So here, for example, I'm giving 1 mark, I'll give you a mark for saying potassium channels are involved. Maybe half a mark for that because you've already got a mark for potassium channels there. But they close a bit slowly. But of course, this is one answer. If you come up with additional information about sodium potassium ATPases, then you get a mark for that as well. So that's where you get your 4 marks there. How about this one? So this is probably a classic short answer question. Describe what is happening in an important process in somebody's lecture. So describe the process whereby a neurotransmitter, such as acetylcholine, is released at a chemical synapse. So you want 4 things. What are you going to say there? Have a little chat, just for 30 seconds, then we'll get moving. So yeah, let's get moving on a bit. So what type of answer are you going to give me there? I think the easiest thing is probably just to show you the diagram. Hopefully this is in your mind. So this is a chemical synapse. And what I told you in the lecture was probably 3 or 4 pieces of information. So I told you about neurotransmitters being released when an action potential comes in. Maybe there's a couple of marks there. Vesicles fuse with the membrane. A couple of other things you could have said. Remember, there's 4 marks maximum. So we want 4 pieces of information. Neurotransmitter acts on receptors, and that's called synaptic transmission. And actually, if you look at my model answer, all I've done really is I've translated those bullet points into a marking scheme. So you see here things like calcium channels I haven't put on the marking scheme, but if you were to include those, that'd be another alternative way to get a mark. So I think the reason I put this one on here was because it illustrates that diagram that's come up 3 or 4 times, or 2 or 3 times in your lectures. If you know that, that's kind of key information for the neurophysiology. Okay, so we've got about 20 minutes left. Let's repeat what we've just done, but with muscle physiology, as opposed to neurophysiology. So these are the lecture aims. Probably the thing to point out here is that we've got 3 types of cell, contractile cell, 3 types of muscle. So we've got skeletal, cardiac, and smooth. So key learning aim, as you'll see as we go through these, structure and function of skeletal muscle, excitation, contraction coupling of skeletal muscle, something called the sliding filament model, and to describe the mechanical properties of skeletal muscle. If we look at the lecture aims for the other lecture, which was on cardiac and smooth muscle, understand cardiac muscle function, understand smooth muscle function, excitation contraction coupling, to be able to compare and contrast contractile cell function. Okay, so let's give you 2 or 3 MCQs, and then we'll give you a few short answer questions and see how we get on. So in the sarcomeres, the thick filaments, so this is all about excitation contraction coupling, but it's asking in a little bit of detail. But basically it's asking you, what are the thick filaments called? Have a chat, 30 seconds. Are they actin? Are they myosin? Troponin? Tropomyosin? Or are they T-tubules? So what do you think the answer to this one is? B, myosin. Anybody for anything else? I think I would agree. And we're right. Good. So, you know, you might have looked, when I read it out, you might have kind of thinking, well, what are sarcomeres? Again, I think basic knowledge here, if you know what the thick filaments and the thin filaments are, if you know one is actin and one is myosin, then that's the type of question we should be asking you at part 1 level. So I think most people knew that. Good. And we've got various, you know, lots and lots of slides on which ones are the thick and which ones are the thin. Just highlighting some information here. Thick filaments are made of myosin, thin filaments are made of actin. So a question about excitation contraction coupling. Again, this might be a little bit more tricky, but because excitation contraction coupling is one of our key learning aims, you should know all about it. So which one is it not typically dependent on? Bit more tricky, but again, maybe you can get to this one by ruling out the ones that are correct to get to the odd one out. Have a chat. Again, this is a bit of a deliberate question. Which one do you think is incorrect? E, someone's saying, I think. Anything else? What are you thinking there? Anybody want to give me an answer? Say again, sorry. So I can't hear because it's quiet. Say again. Just someone shout. Someone put their hand up and shout out. D. Okay, so I think that's what people are saying. Sorry, it's a bit low level noise up at the front there. So we're saying D. Let's have a look. D is correct. Good. So I think for that one, again, it's one of these questions where the other 4 are correct. So you should be, whether the activation of the calcium pump on the T-tubules is a little bit difficult to know. You should know that in order to contract a muscle, you definitely need an action potential. You should know that you need a release of a neurotransmitter. You know, you might have thought that we were trying to trick you by putting the wrong neurotransmitter in. We shouldn't really do that. You know, we could have said release of the neurotransmitter GABA or noradrenaline or glutamate. I thought that would have been a bit mean. So release of the neurotransmitter acetylcholine, we should know that acetylcholine is the correct transmitter. Do we need an increased concentration of calcium to get contraction? Yes, we do. Do we need, let's go to the wrong answer, E. Do we need actin-myosin complexes to form to get muscle contraction? Yes, we do. Key part of the sliding filament theory. So the incorrect answer, the odd one out, would be D, because we don't necessarily need activation of the calcium pump on the T-tubules. We might need activation of calcium pumps elsewhere, but that's the incorrect answer. Okay, and that was a slide that gave you some information about excitation contraction coupling in skeletal muscle. So here's a general one again. We'll see in a minute why I've put this one in. Which are the muscle fibers that are predominantly used to signal something called proprioceptive information? So are they A alpha, A delta, B, C, or D fibers? 30 seconds. So if you remember proprioception, when I'm standing up, proprioception, my muscle spindles and tendons are telling me I'm standing up. So I'm activating some of these fibers standing up. When I go and sit down, I'm using different muscles. I'm relaxing. I'm sat on a chair. I'm using probably different fibers. I'm maintaining my posture with some of these fibers. But which are the fibers that are used in proprioception? What do people think? You want to tell me? Have a guess. I shouldn't say guess, should I? Have an educated. What do we think? Who doesn't know this one at all? So someone says A at the back there. So we've got one answer. We've got a vote for A. The correct answer is A. Well done. Okay, so why did I give you this question? Why did I give you this question? I'm beginning to wonder. Now, so here is a table that's got loads of information in it, yeah? So when I'm writing a question, I can think, right, I've given them this table. There are 5 different things in the table. I've got to ask a multiple choice question. What is key information from my learning aids? I want you to know about proprioception. I want you to know that A alpha fibers are involved in proprioception. I could have asked, you know, which ones are involved in pain. We'll cover pain later on in the course. Bit tricky for pain because A delta and C fibers are probably equally involved in pain. I could have asked you which ones are the most heavily myelinated. That might have been a good question because myelination is one of our key learning aids. I could have asked you which one conducts the quickest or the fastest, those that are heavily myelinated. So I could have asked you lots of things, but I chose to ask you something so that you can demonstrate that you knew about this table, and some of you did. So I should go through this one quite quickly because we've kind of moved away a little bit from this type of question. Leaving blanks in questions, the GPHC don't like that. So we used to do these type of questions quite a lot when I put these slides together, but it's useful. So I think let's just try, as time's moving on, let's just try and go through this. So the 3 major contractile cells, the 3 types of muscle are what? Cardiac, smooth, and skeletal. Good. So we should probably be out. So if you're answering this question, if you get half a mark for each, you should get 1.5 marks really easily. Only one of these shows no clear striations, stripes. Have a guess. Smooth. Good. So you've got 50% already. Which one is the only one under voluntary control? Skeletal. Good. Of these, one of the muscles has spindle-shaped cells, a single nuclei. Another has elongated cells and multiple nuclei. So that's a little bit more difficult, I would say. I think you should know that smooth muscles don't have striations. I think that you should know that skeletal muscle is under voluntary control. But maybe it's difficult to know which one has a spindle-shaped cell and which one has elongated cells. But those of you who are going to get first class will get this bit. Crucially, I'll give you the answers in a minute. The picture above is of what muscle? Skeletal. Good. So let's see how many we got. Like I said, we moved away from this type of question a little bit, but I've left it on just to tell you about other things. So like I said, I think, you know, most people there shouting out the answers at least got 3 out of 4. So 75% on this question. I heard people say smooth muscle, and I heard people say skeletal. I heard a few people say other things. But these are a little bit more difficult. So let's have a look where we got that information from. And again, it's another table. So this table, like I said, you know, this type of information here is a little bit minutiae. But this is the kind of key thing. Voluntary. We should know that you know, if I want to move my arms like this, I'm thinking about it. If I've got smooth muscle, which is in my guts, or cardiac muscle in my heart, I'm not consciously thinking about digesting my food or keeping my heart going. That's done automatically. So key things you can find in this table. And of course, in the other slides, we give you a little bit more information. So this is another table where we give you lots of detailed information. Again, this is a table where we probably wouldn't be expecting to learn all of this, but this is something that supplements the diagrams in the rest of the lecture. Okay. And again, you know, the learning aim number one was to know the difference between these three different types of muscle, contractile cells. So you should be able to identify this one, skeletal, smooth, cardiac. Good. So again, you know, these are the very basic things. So let's say, you know, you're revising and you're thinking, oh, this muscle physiology, there's so much information here. Actually, the kind of questions that we're asking, if you can tell the difference between these three, which is learning aim number one, you know, you can pass that question. You can get, you know, you can get whatever, 2 out of 4, 3 out of 4 on the question, just by basic knowledge. But maybe you need a little bit more in-depth question knowledge to get a first-class answer. So, briefly describe the sliding filament theory for muscle contraction. Again, key learning aim, know about the sliding filament. So the sliding filament theory, called here, or the sliding filament model, is this idea about how actin and myosin interacts with each other. So again, because time is moving on, this is that diagram. So key learning aim, know about the sliding filament model. This is the one slide on the sliding filament. There are 4 stages. So you can predict us being, you know, lazy lecturers or whatever. If it's got 4 stages, we've got to ask you a 4-mark question. Let's ask you about what happens in stage 1, 2, 3, 4. And we'd be expecting you to be able to describe how the actin and myosin interact with each other, but probably at a very simple level. If you read this, it tells us about, you know, there's a power working stroke, and ATP is dissociated, and that provides the energy. We don't probably need it in that much detail. Something about sliding towards the M line. So things like this M line and the Z line, they're going to be too detailed to ask you in the examination. But what we do want to know is maybe that actin and myosin interact. It's triggered by ATP. Calcium is important. And one of the other slides that I gave you actually tells you the things that are important in sliding filament theory. And essentially, you know, if you couldn't remember that diagram, I gave you this memory aid, this mnemonic called ACT4. So A stands for ATP, C stands for calcium. There are 4 things beginning with T, which is tropomyosin, troponin, and thick and thin filaments. So thick and thin filaments fits nicely with our mnemonic, but maybe, you know, if you'd have said actin and said myosin, you'd still got the marks. So a little aid. So if you couldn't remember that kind of complicated diagram, but you could tell me all of those things, you'd have probably got enough marks to pass the question.
So in that question, would it be okay if we said that if we mentioned that those things are needed, or would we have to explain what?
Yeah, so I'd probably, let's say that's a mark out of 4. I'd have probably given you 1.5 marks for knowing the 6 things. And then if you'd described how they worked, you get up to 4 marks. Okay, that's time up, but just bear with me. So what we've also done here is I'll put this session up on Blackboard. There are a bunch of other questions about homeostasis and body fluid. Practice those ones as well, okay?